Aló. Ok, buenos días. Gracias por estar presentes en esta ocasión tan especial y tan significativa para la Universidad de Atacama, de encontrarse entre dos universidades y estar dentro del plan de internacionalización, ya que la Universidad de Potsdam con la Universidad de Atacama ya tiene un convenio hace más de un año. Así que les doy la bienvenida en nombre de nuestro vicerrector académico, vicerrector de investigación y posgrado, el señor Hugo Salinas, nuestro director de la Dirección de Innovación, Desarrollo y Transferencia, señor Tomás Santibáñez. Agradecemos la presencia de CONAF, representado por el señor Caravante, muchas gracias. El decano de la Facultad de Minería, el señor Dante Carrizo, y los investigadores de CRIESAT. Muchas gracias por estar presentes. En esta ocasión estamos con la presencia internacional y expositor senior, el señor Dick Wagner, que es de la Universidad, del Centro Alemán de Investigación de Geosciences, EGFZ, de la Universidad de Potsdam, junto a los investigadores de CRIESAT, comenzaron un nuevo proyecto, una nueva campaña en nuestro laboratorio abierto, que es el volcán Ojos del Salado. Para introducirlos en el tema y contextualizar este seminario, el doctor Rómulo Oces del CRIESAT, botánico, nos va a hacer la inducción y nos va a presentar los seminarios. Muchas gracias.
Gracias, Gladys. Gracias a todos por estar acá. La idea de esta presentación es brevemente hacer un marco teórico de dónde nace esto y tratar desde ahí enganchar con la presentación del doctor Wagner, buscando alianzas y segundo proyecciones, que eso es lo que más nos interesa ahora en esta etapa. Aquí hay un título de la presentación, básicamente esa es la pregunta. Uno podría decir, ¿qué ideas podemos rescatar para poder valorizar y conservar nuestros recursos biológicos en el amplio sentido de la palabra? Y aquí vamos a comenzar a tratar de saber quiénes somos, ¿cierto?, nuestras alianzas, y desde ahí proyectar este trabajo en conjunto con el GFZ hacia los años que vienen. Lo primero que voy a comenzar haciendo es un agradecimiento formal a todas las personas que estuvieron involucradas en esta campaña. Aquí, sin estos socios que han demostrado tener no solamente un conocimiento acabado de la zona, sino que también un soporte logístico que permitió llevar a cabo esto no se hubiese podido realizar. Así que vayan para CONAF, primero como agente administrador. Las gracias por el soporte técnico, el Ministerio de Medio Ambiente también, para poder identificar potenciales lugares. Y obviamente los laboratorios que concursan dentro de la universidad, entre ellos el laboratorio LICA de la Universidad de Atacama y también obviamente a uno de los gestores principales, que es el CRIESAT. Agradeciendo, digamos, a todos ellos, más el concurso del Instituto de Geosciences GFZ de Potsdam en esta oportunidad. Más específicamente, esto, ustedes saben que hay que soportarlo, hacer un apoyo logístico. Están todos los proyectos del Ministerio de Educación involucrados en esta propuesta. En el futuro vamos a tener que plantearnos, ¿cierto?, de cómo podemos aunar recursos públicos y privados para poder continuar esta estrategia. Eso es una pregunta abierta después de esta campaña. Sin embargo, el primer paso es un paso que yo considero grandemente exitoso. ¿Quiénes somos aquí? Hay una serie, en la universidad está ocurriendo algo, está ocurriendo el agrupamiento de gente y de talento, ¿ya? Aquí hemos cruzado los límites del CRIESAT para poder establecer conexiones con otras unidades dentro de la universidad, con otras expertices, con otros talentos. Y eso ha sido también la clave para ir sumando capas de información que van a ser la base de las futuras investigaciones. Ahora, también es importante considerar que hoy día, si hacemos una especie de radiografía a las redes de internacionalización de la universidad, vemos que el ambiente es promisorio. Tenemos socios nacionales, socios internacionales que nos están apoyando y con los cuales tenemos que seguir proyectando este trabajo. Y qué otro lugar más impecable que es Ojos del Salado como un laboratorio natural. Ahora, es importante también indicar de que con el tiempo hemos ido generando una plataforma nueva. Yo quiero traer el primer concepto acá, que es la plataforma de biogeociencias para Atacama. Ese fue un soporte muy potente que parte de estos proyectos ATA del Ministerio de Educación, ¿cierto? Más otros, el concurso de otros proyectos, los cuales de a poco hemos ido armando un laboratorio especializado en este tipo de investigación. Por lo tanto, la plataforma de biogeociencias para Atacama es uno de los objetivos que tenemos en mente de ir concretando ya, no solamente dentro de la universidad, sino que con actores públicos y privados. Bueno, lo que hacemos básicamente en el CRIDESAT es esto, investigar para poder aplicar. No podemos gestionar nada que no podamos conocer. Y este es el primer paso en una serie de proyectos que tienen que ver con conocer en detalle los recursos biológicos de Atacama. Es importante, y ahora con esto voy a ir cerrando, dejar encima de la mesa dos conceptos importantes. Chile es un laboratorio natural. Atacama es dentro de estos laboratorios un laboratorio muy especial, pero este eslogan, ¿cierto?, que suena bien, requiere de un trabajo y proyectar este trabajo. Requiere conservar, requiere articular para conservar y estudiar. Entonces ahí, por ejemplo, es clave ir manejando los conceptos de cómo valorizamos estos recursos biológicos que están ahí, que la palabra clave, que seguramente Dirk también, el doctor Warner va a comentar, es lo único, lo singular que tienen estos laboratorios. Y cierro con este concepto, laboratorios naturales para Chile. Este es un concepto que ya está encima de la mesa y que tenemos que apropiarnos con todo. Nosotros estamos en medio de un laboratorio natural, de un conjunto de laboratorios naturales, no es solamente uno. Nosotros estamos recién comenzando con otras unidades dentro de la universidad a explorar esto. Pero esto hay que considerarlo y tomarlo en primera posta. ¿Por qué? Porque Chile entero es un laboratorio, Atacama también es un laboratorio natural. Y es clave poder identificar estos sectores, empezar a protegerlos para poder proyectarlos. Características que hacen único Atacama, una serie de combinaciones bioclimáticas que pueden ser comparables con la Antártica, con nuestro desierto, con nuestra alta montaña. Por lo tanto, tenemos un mosaico de posibilidades. El poder dar cuenta de la investigación que hay ahí es clave para poder proyectar esto. De lo contrario, vamos a tener, yo diría, un popurrí de investigaciones inconexas, y ese es un riesgo que a lo mejor tenemos que enfrentar desde ya en conversaciones que van a venir después. ¿Qué características elementales tiene Atacama? Es que tenemos todo en una unidad territorial conocida. Por lo tanto, son ventajas muy potentes. Por ejemplo, este gradiente altitudinal es único en Chile. Por lo tanto, ese tipo de aspectos, ese tipo de ventajas hay que transformarlas en ventajas comparativas, ventajas competitivas que nos hagan ser destacados en este estudio. Dejo hasta aquí esta gran pregunta, ¿cómo poder abordar este desafío de poder valorizar? Y uno del primer paso es comenzar estudiando nuestros laboratorios naturales. Muchas gracias de nuevo por venir. Dejo abierta esta pregunta, más que una sentencia, que en parte el doctor Warner va a explicar mayormente. Muchas gracias.
Hello. Good morning. Buenos días. So, these are some of the words in Spanish which I know, so therefore I'm sorry that I have to give my presentation in English. I hope this will be okay. And I also would like to unwear the mask, otherwise it's like on Ojos del Salado, so I hope it's okay. Yeah, so thank you very much, Romulo, for the nice introduction. And I'm very pleased to be here today and give this presentation about the work, mainly about the work which we have done within the framework of the Earthshape program. But before I start with the science, I would like to give a brief introduction into the GFZ, the German Research Center, which was asked by Romulo. And by the way, the title of my presentation of Survival Artists and Superheroes, this should indicate a bit that we are working in extreme environments and that the organisms are really superheroes, that they can survive and be metabolic active at the end in such kind of extreme environments in terms of, for example, moisture or solar radiation. Yeah, this is the view, an air view of the campus Telegraph Hill in Potsdam. So here you see this area, and this is near the main station of Potsdam. And in the background you can see the city center of Potsdam. Yeah, this is a view of our main buildings, but there are some more. The GFZ, the German Research Center for Geosciences, was founded in 1992 under public law. It is the German's National Research Center for Solid Earth Sciences, and it is member of the Helmholtz Society or Association, which is the largest science organization in Germany with up to 40,000 employees. In altogether, I'm not sure, 18 or 19 centers like the GFZ. So some facts and figures. So the members of staff in December 2020 was almost 1,300. Most of them are scientists, mainly located in Potsdam. But you can see there are also some observatories and smaller dependences in Germany. The budget in the last year was almost 100 million euro. 2/3 is institutional funding from the federal government and from the state of Brandenburg, where Potsdam is located, and 1/3 is third-party funding. So the mission of the GFZ is System Earth, from core to space, and the main viewpoint is solid Earth science, as you can see here in this graph. So we are focusing mainly on the geosphere, of course, but in relation to atmosphere, hydrosphere, cryosphere, and biosphere. And you know, maybe I'm a biologist, microbiologist, working at the Geoscience Institute, because it's important to have this integrative and interdisciplinary research. And therefore, we have built a new laboratory, the so-called GeoBioLab, which was opened in the last autumn, and which should bring more closely together geoscience and biosciences, also with new unique laboratories in this building. So my group is sitting there since last autumn and has started to work. Okay, coming to the science part. As I mentioned already, the first part will present results of the Earthshape program, which means Earth surface shaping by biota, which is a German-Chilean research initiative funded by the German Science Foundation. And you can see the main player in this game, let's say. So it's a collaboration between Romulo and me. And we had two PhD students, Lisa Marie Moskva, she was PhD in the first phase of Earthshape, and Victoria Rodriguez, who is sitting over there, is PhD in the second phase of the Earthshape program. And so we are mainly interested to learn more about the impact of microbial communities. We call them here microbial engineers, on the soil development and also the stabilization of soil ecosystems. So the program was also established to overcome the geoscience paradigm that the Earth's surface is mainly driven by climate and tectonic forces. But as soil scientists, we also know that, of course, biota plays an important role. And this is not only the vegetation which you can see, but also the huge amount of microorganisms which live and colonize the soils and also the extreme soils, as I will show you later. But of course, pathogenesis is also driven by climate factors, by topography, but also by the parental material, and it's also a matter of time. So this is an overview of the study sites, and Romulo mentioned this already in his presentation, that the Chilean Coastal Cordillera is a natural laboratory, especially because soil formation took place on the same parent material. So the climate gradient is one factor which influences the soil development and makes a difference here. You see our study sites from arid to humid temperature in the national parks or reserves, Pandazuca, Santa Gracia, which is semi-arid, La Campana near Santiago, which is Mediterranean, and Naalbuta in the central south, which is humid temperature. And the general concept was to take samples from different soil profiles along a slope exposure, and we took samples in different depths, down to 40 centimeters, but also samples of the saprolite material down to 2 meters. So this gives an overview of the soil classification, which was done in the first phase of the Earthshape program. You see that in the desert in Pandazuca, we have initial soils, which we are calling rigosol. So there's almost no humus accumulation and also almost no other soil-forming processes. But this developed along this gradient. You can see we have the so-called cambisol in Santa Gracia, but also in La Campana, and the umbrisol in Naalbuta. And here you can see also the climate characteristics, especially the precipitation, which is only 12 millimeters per year in Pandazuca, but almost 1,500 in Naalbuta. So two impressions of the soils, nice images, which we're taking during the first campaigns and then also used for the publications. So this is the initial soil, the rigosol in Pandazuca. You can see the different horizons here, so there are no big differences, no real soil-forming processes, as I already mentioned. But on the other end of the transect, we have the umbrisols in Naalbuta. You can see that this is a real developed soil with a huge horizon at the top, which is more than 50 centimeters, and which has a content of organic carbon of up to 15%, which is really huge, and this makes, of course, a big difference compared to other soils along the transect, and especially to the desert soils. So this graph shows some trends of the soil physical parameters which were measured in our lab, but also in the whole consortium. There are only 3 examples. You see, for example, the pH, which is slightly alkaline in Pandazuca, decrease slightly to Naalbuta, which is then slightly acidic. You can also see what was also indicated in the soil pictures, that the organic carbon content drastically increase from Pandazuca, so from the desert regions, to Naalbuta. And this is the same trend as you can see here, the DNA amount. DNA is a genetic material which is the base of our microbiological studies. And you can see that also the amount of DNA increase from Pandazuca to Naalbuta. So we have two main research questions. The first one is, what is the status quo of microbial communities in Chilean soils? So how different climate conditions and vegetation cover alter the community, and can we detect predominant generalist and site-specific specialists? And the second question is, how and to which extent are microorganisms involved in soil formation and stabilization processes? And I think this is of general interest if we have in mind global warming, which changed the environment. There are some publications saying that also the Atacama will get at some places more rain in the future. And this means for us it's a question how develop these soils and under these new climate conditions, and which role play microorganisms for the development. On the other hand, we have the developed resilient soils, for example, in central Chile, which are also used for agriculture. And here's, of course, the question, how will they affect by climate warming? And how can we use microorganisms, for example, as early warning indicators, but also to stabilize the ecosystem also under other or different climate conditions? So these are the methodologies which we apply and which we have established in my lab. So 4 toolboxes. The main toolbox is molecular biology, so based on the genetic material which we can extract from the soils and which give us information about the abundance of microorganisms in the soil and the diversity and also the function. We do also a lot of geochemical and biogeochemical analysis because this is necessary to understand the processes and the behavior of the microbial communities. And then some ecophysiology and also the bioinformatics, which is somehow the glue which brings everything together with the bioinformatic analysis and statistics. So some results. Here you can see the microbial abundance for bacteria, for archaea, so these are very similar to bacteria, also single-cell organisms, and the fungi, which is the main part of Romulu's work in this project. And you can also see the diversity index here. And it is obvious that the trend is similar for bacteria, archaea, and fungi, so decreasing, let's say, cell numbers with depth, and also unique pattern for Pandazuka. You see Pandazuka is a little bit different, but nevertheless, for example, here for bacteria, we have still cell numbers in the range of 10 to 5 and 10 to 7, 8, which is really quite high for such kind of desert environment. And the other 3 study sites, they show more or less the same behavior along the transect. So this is the result of our microbial community analysis. So don't worry about this complex graph. The bar charts and the different colors indicate different groups of microorganisms. And you can see here 3 groups of our study site. So we have again a unique community in Pandazuka with a high variation between the depth increments, which you can see here. So this is from top to bottom in the soil. And you can clearly see that there are shifts in the microbial community with depth. So most complex communities with only minor variation of the different groups of microorganisms can be seen here in Santa Gracia and La Campana. And this was quite surprising for us, because if you go to the field, you see that Santa Gracia, it's like a desert, while La Campana is like a Mediterranean forest. And I had not imagined before that these communities can be somehow similar. But you remember maybe that also the soil type is the same, and therefore the conditions are more similar than expected. In Santa Gracia, we think that fog brings the moisture which is necessary while in La Campana there's a little bit more rainfall at all. And Nalbuta again is also different from all the other study sites. It's a stable and less diverse community in this study site. So we have then also analyzed bacterial habitat generalists. So these are organisms which can live in all the study sites, which is also quite interesting because we have so different climate conditions. But nevertheless, we found some organisms which can live in all the sites under the different climate conditions. So at all, we identified 22 different microbial groups that predominate in all 4 climate regions. So this is a core microbial community. On the other hand, we have the habitat specialists, which are organisms which are well adapted to the given environmental conditions, and therefore they exist only in 1 or 2 of the different study sites. For example, here you can see that this group of bacteria exists only in Pandazuka and in none of the other study sites. It is also the case here, for example, for this group, which can only exist in Nalbuta. And you see that again, Santa Gracia and La Campana, they are more similar to each other. You see here groups which exist in both of these study sites, but not in the desert and also not in Nalbuta. And this is quite interesting. We have used these generalist and specialist analyses to establish an index. And you see here that the amount of generalists increase along the climate gradient, while the amount of specialists decrease. And because the generalists are more flexible, more adaptable, this indicator indicates that some of the study sites are not as stable as others. So Nalbuta, for example, is not as stable as Pandazuka. And this has to do with the climate conditions, but also with erosion processes. And this could maybe also help to analyze other ecosystems also under the question of ongoing climate warming. So we have also done soil manipulation experiments in this program in the first phase and also in the second phase, which is the core of the program, I would say, because we can learn a lot of the impact of microbes on soil formation by these simulation experiments. So in the first phase, we did a soil structure simulation experiment. And in the second phase, and this is the main experiment of Victoria, we did a climate simulation experiment. And you see here the more specific research questions related to these 2 experiments. So in the first phase, the main question was what impact microbes have on the soil aggregate formation, which is one of the first soil forming processes, and also a process which stabilizes the soil surface, for example, against erosion. In the second phase, it was more the question, how would a desert soil develop if the climate changed and we have more precipitation, for example, like in Nalbuta? How would the soil develop, and which influence have the microbes on this development? And this graph shows the concept of the soil structure simulation experiment. We have done 6 cycles of wetting and drying. So in general, wetting and drying is performing aggregates in soil. We have done this with the untreated soil, so with the natural soil, but also with sterilized soil samples to avoid the impact of microbes, so to be really able to identify the impact of the microorganisms. And at the end, we have analyzed the starting point, cycle 1, 3, and 6, and then we had also a constant moisture reference. So, and these data are in progress in evaluation, so I can only show you here a first result for the aggregate fractions in La Campana, which is from our partner in Tübingen. You see on top the soil from La Campana, the topsoil, and in this graph with microorganisms, and then in this graph without microorganisms. And the same is here for a bottom soil sample with and without microorganisms. And you can see that there's a clear shift in the aggregate formations if microorganisms are available, as you can see here compared to this graph, or even more in this graph compared to the analysis without microorganisms. So this shows that the microbes have a clear impact on aggregate formation. And at the moment, we analyze which organisms are responsible and in which way. Another result from these simulation experiments is shown here. So the response pattern regarding the abundance, so the numbers of microorganisms. You can see here a negative effect for Nahalbuta. So drought represents a strong stress factor for microorganisms in Nahalbuta, which are usually not used to drought, that potentially lack such adaptations. You can see again a similar effect here for La Campana and Santa Gracia, so we call it a neutral effect. So microbial communities in these two sites are already selected for moisture stress tolerance because they are used to drought. So it's their daily routine, let's say. And in Pandazuca, we have a positive effect, which shows that production of dormant stages until environmental conditions become more favorable in Pandazuca. And if there's enough water, then the organisms can be getting active again. And Romolo also proposed that, especially for the fungi, maybe there is a full life cycle possible under these simulated conditions. Yeah, and if you have a closer look on the microbial communities, you can see that in Nahalbuta, La Campana, and Santa Gracia, the same groups of microorganisms, the same phyla, are active. But if we go deeper into the microbial community composition, we can see that in Pandazuca, there's a unique group responding, and different species are active. Okay, so this is the concept of the climate simulation experiment performed by Victoria. You see that we have used the two desert soils sites, or two desert sites. So, and we have used the A and B horizon for this experiment. And we have simulated the climate conditions from Nahalbuta, so a temperature range of 18 to 11 degrees centigrade, a photoperiod of 14 to 10 days, and a precipitation of almost 1,500 milliliter per year. So we had 4 treatments, the natural in situ samples, and I mean to simulate the soil development, we have also had some biocrusts in one of the treatments. And in another treatment, we have had pioneering plants. And then again, we have had the sterilized control to be able to identify the impact of microorganisms with 3 replicates each, and then 4 sampling points, or I mean the 0, the original status quo, and then 2, 12 and 16 weeks for sampling. This is also under analysis, but I can show you here some pictures from Victoria. So how the soil microcosms look like after 2, 12, and 16 weeks. And you can see, for example, that there's a growth of biocrust. Yeah, you can also see that there's a growth of the pioneering plants. And in the last, for the last period, the plants were cutted to learn more about the effects of this dying material, decaying material in the soil. And the same for Santa Gracia. You also see again here huge biocrust growth and also the plant growth, and then again the plants were cutted for the last treatment. Yeah, but as I said, so this is under evaluation. So Victoria has analyzed the soil microbial communities, and now we are waiting on the results of our partners. So then we have to bring together everything and can continue the analysis. So the summary for this part, so we have seen that the climate gradient and slope exposure influence bacterial community diversity and structure in interactions with soil physical and ecological properties. The bacterial community structure changed gradually from pioneering organic matter accumulating communities in Pandazuka, which was quite interesting in comparison to Nalbuta, where we have an organic matter decomposing community already. And we have also seen that extreme conditions promoted greater bacterial specialization in Pandazuka and Nalbuta along the climate gradient. So in the second part, I would like to present some data from the Atacama near Antofagasta. So this was research done in a project of a colleague from the Technical University in Berlin, where we were involved. And this was related to learn more about the limits of life in such kind of environments. And I mean, if you see here a picture from the Atacama, so if I asked you how many bacteria are living there, you would maybe say 0, but this is actually not true. And I will show you that these bacteria can be even active under the extreme conditions of the Atacama Desert. So as microbe there, you have to cope with extreme environmental conditions. For example, no water, and intensive UV radiation, and also high concentrations of salt, like the perchlorates, for example. And these conditions are more intensive in the high altitude, and this is what we have learned during our campaign to Barrancas Blancas, the Ojos del Salado site. So this was really hard also for us, extremophilic humans, so to work under these extreme conditions. And this is, of course, also interesting in comparison to life beyond Earth. So, for example, on Mars, you see, if you have a comparison of the images, so it looks quite similar, and you know that also on Mars there are quite high volcanoes, the highest volcanoes in our solar system. So everything which we can learn from the Atacama is also relevant for these astrobiological topics. So for this research, we have taken samples also along a moisture gradient from the coast to the core of the Atacama here in Yungay. And then there were 2 more study sites, hyper-arid study sites in Lomas Bayas and Maria Elena. We have here applied the first time a new DNA separation method. So I will not go too much into the details, but usually if you extract the genetic material, you get all of this. And you don't know which of the organisms are still alive and doing something, and which DNA is from dead cells. And this makes the interpretation of your data complicated, and therefore we have established this method where you can differentiate between intercellular DNA, so the DNA from intact and living cells, in comparison to the extracellular DNA, which represent, let's say, DNA which is preserved naked outside living cells in the sediments. And we have established this method already 2014, but nowadays, Diego Medina and Lucas Hausmann, also sitting here in the audience, they have started to improve this method, especially for specific environmental conditions, and this publication is also in preparation. So to show you what the difference is between the total DNA extraction and our new method, I would like to show you this cladogram. So this shows the composition of the microbial communities in a soil sample from the Yungay Valley. And the same picture if you extract E and I DNA. You see that all the blue lines here is eDNA, which is from dead cells, let's say, and iDNA, the red one, is from living cells. And if I show you this picture, you will see that a huge amount of the organisms which seems to be alive in this graph is actually not anymore existing in the environment. And on the other hand, we have, of course, also living organisms, but it shows that there is a huge difference. And if you are interested to learn more about the living organisms, you need to separate it, and you see that the picture which you get is quite different from the total DNA extraction results. So again, some data on the microbial abundance in the desert soils in the Atacama. You see here the separation of E and I DNA in blue and red. And then we have also done, in cooperation with a colleague from the GFZ, the so-called fatty acid analyzers. So these are also biomarker indicators for living organisms. And you see that we have a good comparison between the eDNA, which is the living community, with these biomarker also for living organisms. And in general, we have also relatively high abundances here between, let's say, 10 to 3 or 4 up to 10 to 6, something like this. So also these extreme hyper-arid areas are colonized by living organisms. And we have here had a look on the top 50, let's say, species to see which are more relevant, and also to see if there are differences between the E and I DNA pool. And I will also only give you some impressions here. For example, 2 organisms from the Actinobacteria group. You see that they are prominent here, for example, in the red sand site. They are adapted to dry conditions and also high salinities, and that you find them in both DNA pools indicate that they are really alive and that there is a turnover. And this is also true for the Halobacteria. So they are adapted to high salinities, and you find them especially here in the coastal soil site, which is affected by the sea spray and by the fog, and therefore this site is characterized by higher salt concentrations. And then we have also the Bacteria, which are organisms which can form resting stages to survive under extreme conditions, but you can see again here they are highly abundant in one of the sites in both pools, which shows that they are alive and being active. And in this consortium, a lot of other analyses were done, so I will not go into the details, but it was something about the ATP content, which is the energy currents, let's say, of microorganisms, then the metabolites in the cell, and also a new method of genome replication. And all of this shows that the organisms can be active at least from time to time if there is moisture given by fog, for example, so that even the hyper-arid sites can be a real habitat for living organisms. So to sum up this, the iDNA pool improves our understanding of the living part of the communities, and this would be also interesting, of course, in the framework of the whole Earthshape program, to learn more about the living organisms along the climate transect, but also their response to changing environmental conditions. The eDNA pool provides eventually insights into microbial community changes in time and space and response to changing environmental conditions, so it can give us also information about the past microbial communities. And the living community is dominated by specialists best adapted to the harsh environmental conditions in the hyper-arid Atacama Desert. Yeah, and with this, I would like to acknowledge the funding organization, so for the Earthshape program and also for the project of the TU Berlin and all the partners within our Earthshape program. And yeah, the funding organizations, German Research Foundation, ANIT, Becker-Schiele, and also CONAF for their support in the national parks, which was very helpful for our work, and also with all this permitting and all this bureaucratic stuff. So thank you very much for this. And with this nice image from Romulo and Mir from the Barrancas Blancas side, I would like to thank you for your attention.
Agradecemos un seminario realmente que realizó el doctor Dirk. Dejamos abierto el micrófono para alguna consulta, dudas.
Yeah, thanks a lot for the presentation. Yeah, and one question, according to the fact that in Pan de Azúcar, so apparently there is mainly specialists. But according to the experiments you made with drying and wetting, this is the Pan de Azúcar community which tend to have the best adaptability, no? So does it mean that specialists, a community of specialists, have a better adaptation capability to environmental change?
I mean, specialists are well adapted to the given environmental conditions, and it indicates stable conditions because, I mean, they are specialists. If the conditions would change, let's say, in a yearly interval or so, they had problems to survive. I don't know if this answers your question.
Apparently.
No, it's okay. Yeah, so I mean, you know, we have not only the specialists, as I have shown, we have also the generalists. So we have a larger pool of organisms, and they respond in one or the other direction to these environmental changes. So if we add water or we apply these wet-dry cycles, then this will have a positive impact on one part of the community, for example, also on the fungi, but it will have a negative effect on others, maybe on the specialists. But this is something, as I said, so the data are not fully evaluated yet, but this is what I would expect.
Well, thank you very much for your presentation. Yeah. Yeah, I would like to go to another point. Maybe it was a matter of our discussion in the previous day. According to your experience, considering your previous result in Ojos del Salado, so what are the main recommendations to conserve or study these unique places?
You mean in terms of organization, or?
Yes. In terms of organization, and to how to preserve these places for future investigation. Yeah.
I mean, I think this is a unique study site, let's say worldwide somehow, because it's good, good reasonable by cars and so on, and provide, yeah, an excellent natural laboratory for these high altitudes. And so we were there to study the impact of a temporal lake, and we proposed that this is a hotspot for the microbial communities because they depend on moisture, and this moisture is given by this temporal lagoon. But in general, what my experience is from my work, previous work at the Alfred Wegener Institute for Polar and Marine Research, you need to coordinate the science somehow. Because otherwise you will destroy this unique natural laboratory. And I mean, if you have stations in the Arctic or Antarctic, there's a clear procedure that you have to submit proposals for your planned research, and then there's a committee, a board, which is evaluating your suggestion, and then you get the go or the no-go. And this means that there's a control about the research, who is going there, what is he doing. You can also implement environmental standards, something like this, to ensure that the area is still protected against too much human influence, let's say. So this is what I know from these kind of environments. And I mean, this is more or less the same. I mean, you plan also this high altitude laboratory, and this would be the base for all the science in this area, and therefore I would suggest to follow these standard procedures which are established for the Arctic and Antarctic.
¿Alguna otra consulta? ¿No? Agradecemos la participación del doctor Dirk en este seminario. Agradecemos la colaboración de todos sus estudiantes de doctorado, el apoyo de CRIESAT y del LICA a esta campaña, y los invitamos cordialmente a un brunch. Así que, por favor, los invitamos a todos a pasar a disfrutar un refrescante brunch. Adelante.